Hello everyone, my name is Alexander Belagor, and I did my project on the mitochondria and the mitochondria's role in aging and disease as we get older. So my goal and outline of the project, statistics on aging are pretty impressive. 100 out of 100 people get older. Why does this happen, and what are the biological processes responsible for mammalian aging in humans and mice, as I will further elaborate later in the project. But in this project, I'll explain in detail how and why we get older and the role that the mitochondria plays in the aging process. Some of what I will be covering in this presentation includes how do we age, why does it matter, how the mitochondria keeps us functioning, a little bit of review in these first two slides, what goes wrong, why do we age, the diseases that come from aging in the mitochondria, and a conclusion. Why does it matter? Everyone knows, somehow, that the mitochondria is the powerhouse of the cell, but not many people know that the mitochondria also plays a role in the aging process. In this project, I'll explain what we already know about the mitochondria, clarify what it means to be the powerhouse of the cell, and connect it to the aging process. Hopefully, my audience will be able to take away some of the unique and often unheard of functions of the mitochondria, so that the next time someone jokingly brings up the cliche powerhouse of the cell line, they can be formally lectured and educated on the properties of the mitochondria. Why does it matter? As we get older, we are more prone to aging diseases. I'm sure that everybody knows a grandma or a grandpa who has, unfortunately, some aging-related diseases. These can include muscle weakness, inflammation, bone frailty, immune suppression, and also some pretty nasty diseases like osteoporosis and other spinal diseases or bone density related diseases. In all of these diseases, however, the deterioration of the mitochondria plays a crucial role in the development. So a little bit of review in these slides. The general mitochondrial function, which we went over in class a little bit ago. How do things work before deterioration? So I have a pen here, let's see if it works. Most of the ADP or ATP, or a lot of it, occurs in the oxidative phosphorylation stage, where the proton gradient is established, and the ATP synthase, this little motor right here, is spun, and basically it takes electrons and binds them to hydrogen, establishes the proton gradient, and water is made as a byproduct. So it's important to remember the significance of the ATP itself, and that most of the ATP produced in our bodies comes from the inner membrane of the mitochondria during oxidative phosphorylation. And actually what the ATP does is it prevents us from aging. So without the mitochondria, we would not have the energy needed to survive and maintain homeostasis. It cannot be overstated how much we rely on the ATP for all of our different functions, which include cell signaling and repair. Without ATP production, our bodies would not operate, and we would probably just shut down because we don't have energy. Again, disastrous consequences. It would make sense that without the crucial ATP needed to maintain homeostasis, some of our bodily functions would lag behind. This is a little bit of foreshadowing. So what causes aging as seen in the mitochondria? Mitochondria actually have their own DNA, which scientists have conveniently labeled mtDNA for short. It makes the rest of this presentation a lot easier, which can be damaged like DNA. So just like our own DNA, which is located in the cell nucleus, our mitochondrial DNA can be damaged, and damaged DNA is really, really bad. Damaged DNA can end up encoding or translating for broken proteins that harm us and alter biological processes. So it can mess up the functions of proteins. mtDNA damage can greatly reduce the production of ATP in the mitochondria if it damages proteins like the ATP synthase, which are responsible for oxidative phosphorylation. And both the DNA in the cell nucleus and mitochondrial DNA have repair mechanisms. However, the DNA in the mitochondria is much harder to repair, and the repair mechanisms are greatly inefficient compared to the regular DNA repair mechanisms. So a little bit about how the mitochondria DNA gets damaged, primarily by reactive oxygen species or free radicals. And I have some photos of some free radicals up here. So free radicals come from oxygen, which is again very electronegative when it's used during aerobic respiration as the terminal electron acceptor. Oxygen free radicals are produced. This happens when the electron transport chain, which is supposed to ideally deposit electrons onto oxygen, reducing them into water, fails to create water and makes a superoxide radical instead. And that's again denoted by this symbol and by this equation. It's estimated that reactive oxygen species are created for every 0.1 to 2% of electrons passing through the transport chain. So again, these ROS are very harmful to the DNA, and cells usually make enzymes to detoxify the free radicals, which make them stronger and more resistant, kind of like our immune system. The more we beat at it, the stronger it gets. Usually, we're kind of like studying for a test. The more you study for it, the better you'll be. But too many free radicals can damage mitochondrial DNA, kind of like how too much studying can make you stressed out and burn you out. But the reactive oxygen species can damage the proteins that would otherwise control the reproduction of the mitochondria, and end up introducing additional errors into the mitochondrial copies. Cells usually contain hundreds of mitochondria, but some of the errors that occur can make the dysfunctional mitochondria that are created survive longer than healthy mitochondria. And since the DNA is harder to repair, the dysfunctional mitochondria buildup can become exponential, and the broken ones can become more common than the healthy ones. So an example, this is going to be my primary source. Some scientists did an experiment on mice, and they mutated some of the DNA in these mice, obviously the ones that don't look as healthy, excluding this one. But the mutator mice were given point mutations in their DNA by, in quotes, expressing the mitochondrial DNA polymerase with an altered catalytic pelage subunit that contained alanine instead of aspartate. And what they noticed, here's a cross section of subcutaneous fat where the wild mice and the mutated mice, the wild mice had more fat here at 25 weeks and 40 weeks than the mutated mice did. And that's just one of the findings of their study. For diseases related to aging, the mice with the DNA mutations had reduced fat content, reduced bone density, reduced whole body bone mineral content, higher likelihood of osteoporosis, lower hemoglobin concentrations, an enlarged lumen on the left ventricle of the heart, and a reduction in fertility. And there are some graphs to back up their findings. So, to conclude, like all mechanical engines in the real world, mitochondria are prone to fail at some point. As it turned out, mitochondria failure is due to, in part, high levels of damage from hard to repair, or too hard to repair mitochondrial DNA by free radicals in the form of reactive oxygen species. And the damage to the mtDNA severely reduces ATP production, which is again very crucial, and leaves little energy for maintaining homeostasis. So knowing why they fail can obviously help us to come up with a solution, because the first step in solving a problem is finding out what the problem is. Here are my sources.